Welcome back. My name is Sanjana and today I'll be explaining Chapter 6 of the Brain Facts book. This chapter is called The Developing Brain. So let's get started into induction. Neural induction is when signals from the mesoderm trigger cells from the ectoderm to become nerve tissue. So one example of a signaling molecule is the sonic hedgehog, which is secreted from the mesodermal tissue lying beneath the developing spinal cord. So the fate of the developing cells is its proximity to the signal. So a high concentration turns the cells into glial cells, a lower concentration turns them into motor neurons, and a very low concentration turns them into interneurons. So the signaling pattern is similar in humans and in flies. The next stage is proliferation. Proliferation is when the stem cells and progenitor cells eventually turn into neurons and glial cells when they undergo a series of divisions, which increases the number of cells in the brain. As these divisions progress, the cells begin to divide asymmetrically. One daughter cells keep proliferating, but the other one reaches their ultimate cell, such as a neuron or glial cell. Protein defects from symmetry to asymmetry divisions can cause microcephaly, like a smaller brain, and the excessive proliferation of brain cells can lead to megancephaly, the opposite. 3 to 4 weeks after a human baby is conceived, migration happens. Migration is when a cell moves to a long-term location in the brain. Continuing to divide, a flat neural plate grows and is followed by the formation of parallel ridges. Then the ridges fold toward each other and fuse into a hollow neural tube in week 7, and they move radially and parallelly to the brain surface. 90% of the migration in humans happens with radioglia, which project radially. So these move parallelly to the brain surface. The first ones to migrate are the oldest and deepest layer of the cortex, and the last ones to migrate are the youngest and newest layer. Exposure to alcohol, cocaine, or radiation can prevent proper migration, leading to epilepsy in the child. Making connections depends heavily on external experiences after birth. Axons, presynaptic, produce and transmit signals to other neurons, and dendrites, postsynaptic, receive signals. The longest axons reach multiple times their own size. Developing axons grow by the extension of their growth cone, and enlargement of the tip of the axon which helps finding its precise location. Receptors on the growth cone enable its responses to these environmental cues. So signaling molecules include families of proteins with names such as netrin, semaphorin, and ephrin. These signaling families were first discovered in flies and worms. Synapse formation. The formation of synaptic connections would be highly specific. Both processes, presynaptic and postsynaptic, ensure a synapse can transmit signals quickly and effectively. Astrocytes exert their own influence on synaptic development and function. A single astrocyte can contact thousands of synapses across multiple neurons. So an array of signals determines which neurotransmitter a neuron will use to communicate. For example, motor neurons only use acetylcholine, whereas in a culture with no other neurons, a neuron is predicted to produce norepinephrine. Myelination. Myelination is the fatty wrapping of axons by extensions of glial, and this increases the speed at which neurons can carry signals by 100 times. So, nodes of Ranvier are regularly spaced gaps interrupting the sheath, and this is called saltatory conduction, where the signal jumps from one node to the next, resulting in a faster transmission. Paring back and synaptic pruning. So about half the neurons generated during development survive to function as adults, so half don't. And trophic factors, less sustaining chemicals, are produced in limited quantities, and they are very important for the survival of neurons. And one very important trophic factor is the nerve growth factor, which is important for the survival of sensory neurons. So these cells initially form excessive number of connections, and pruning back is heavily dependent on the relative activity of each connection. So adult brains are formed by pruning away the incorrect connections, leaving only the correct ones. That sums up chapter 6. If you haven't yet, please check out my other videos on the last 5 chapters, and I hope you're able to join me on chapter 7, The Infant, Child, and Adolescent Brain. Thank you for watching.